So every string or read that we sequence come from the much larger string of the genome. So one can be interested in obtaining the superstring that is the source of the genomic in the organism. So by using different techniques we can get the superstring, and the one simple technique is we can combine the substrings to obtain the superstring. But there could be different sizes of superstring, and one could be interested in obtaining the shortest possible superstring, and by obtaining that shortest superstring we can say that this is the first approximation sequence of the longer genomic sequences. To obtain the shortest string we have to use some algorithm, and this algorithm is known as the shortest superstring problem. And the problem statement is that if we are given a set of strings, we have to find the shortest string that contains all of them. That means that we have to find the superstring that contains all of the substrings. So for this we have to give the input to the algorithm, and the input will be the strings of different sizes S1, S2, S3 and up to the Sn. And the algorithm will process and it will give the output in the form of a string S that contains all strings S1, S2 and Sn as substrings. So that string will be the superstring, in such that the length of S is as small as possible. So this superstring S that contains all the substrings S1, S2 and up to the value of Sn should be the smallest in size as much as possible. The figure presents two superstrings. We can see here this is the one superstring and this is the second superstring for the set of all 8 3-letter characters. These are 8 3-letter characters, strings in 0, 1 alphabets, because we can see there are only 0 and 1 alphabets in different arrangements and in the formation of 3, and there are total 8 3-letter alphabets. The first superstring is obtained. This is the first superstring, and by simply interconnection of all 8 strings, we have seen that these are 8 strings, and if we simply connect them we will get this superstring, and we can assume that this is one of the fragments of DNA, the second is second fragment of DNA, third, fourth, we assume these as a fragment of DNA, and if we join them, combine them together, then we will get the whole sequence of the long genome. So this is the one technique we can use and we can get the superstring. While the second one is the shortest superstring, we can see its size is much smaller as compared to this superstring, and this superstring we can obtain by using these 3-letter codes. For example, we can see here that this 000 is the first 3-letter code, and then we can shift 1 just like a sliding window, and then we will use the second set of the 3-letter code, that is 001, and we know that there is a 001 here. Then we will slide another one and we will start from 011, and we know that we have a 011 here. Then we will again slide one more and we'll start from 1, 1 and 1, and we know that we have this letter code 111 in this position. and so on. So in this way, we will use all the 8 3-letter combinations 1, 2, 3, 4, 5, 6, 7, 8, and we will get a very short superstring. On the other hand, we have also used these 8 3-letter alphabets as a substrings, and we have get the larger superstring. So the strategy is different in both of these. So we can say that the shortest superstring in this case represents a solution of the clever theft problem, and it is the minimum string of test a thief has to conduct to try all possible k-letter passwords for the combination lock. So in this way we can get the shortest superstring, and this shortest superstring explains all of the substrings that is present in the superstring. So if we define the overlap SI/SJ to be the length of the longest prefix of SJ that matches the suffix of SI, the shortest superstring problem can be casted as a traveling salesman problem in a complete directed graph with n vertices. That n vertices corresponding to the string starting from S1, S2 up to the Sn, and edges of length that overlap Si and Sj. We can see in this figure that these are the 8 3-letter alphabets 1, 2, 3 and so on, and they overlap with each other. And this reduction of course does not lead to an efficient algorithm, since the traveler salesman problem is NP complete. Moreover, it is known that the shortest superstring problem is itself NP complete, so that the polynomial algorithm we have discussed earlier as NP complete for this problem is unlikely. The early DNA sequence algorithm used is simple greedy strategy.